Everyone. So this is our virtual lecture. Um, if you have any questions, as always, feel free to email me, but we're just going to jump right in. So to quickly review what we started talking about on Tuesday. Oops. Let's go ahead and present this. So obviously we've been talking about photosynthesis. We were talking about modes of nutrition and kind of just the names that we give to organisms to describe how they get their energy or how they get their starter molecules for them to make energy. So we have a heterotroph, which is the other feeder, meaning that they require things from other organisms. This is an example of most of the animals that we think of. House pets, humans, animals that we consume for nutrition, stuff like that. Next, we have the autotroph, which is a self-feeder, meaning they make their own organic molecules. And then we have a photoautotroph, which is what we normally think of when we think of like plants. These are organisms that are able to do photosynthesis, meaning that they're able to take fixed energy from the sun and transform it in and organic molecules and transform it into the energy that they need. So again, these are just some different types of photoautotrophs. And then we started getting a little bit into some terminology. And we talked about how leaves are the main site of photosynthesis. Inside the layer of the leaf, so if we were to take a cross section, we refer to that as the mesophyll. And this is that tissue layer inside the leaf with high concentration of chloroplasts. So if you remember, chloroplasts are kind of like these little discs, and they have an outer membrane, an inner membrane space, and an inner membrane, meaning that they are a double layer membrane organelle. Then we have the thylakoids, which refers to one of those tiny little discs and the thylakoid space. And then we have the granum, which is the kind of stack of the thylakoids. And we have the stroma, which is just the interior space of the chloroplast. So inside of our thylakoids, we have the chlorophyll, which is that green pigment that absorbs light. And if you remember, the reason that we see it as green is because green is the color of the electromagnetic spectrum that goes through the chloroplast and is kind of comes back out of the chlorophyll. It's the type of light that's not absorbed. So if you remember from our last conversation, the majority of light that's absorbed by chlorophylls, A, B, and carotenoids falls in like that 425 to 475, 490 wavelength. And you can see that here where those peaks are. Here where the green wavelength is, there's almost no absorption, or there is no absorption from the carotenoids happening. And then back again in this red light, red area of the spectrum, we have some more absorption from those chlorophyll A and chlorophyll Bs. So then we have, sorry, just to finish up, I guess, our terminology. We have the stomata, which are the pores in the leaf. If you remember from the last video we watched with the cacti, this gives plants the ability to kind of open these pores and allow carbon dioxide to enter and oxygen to exit. And then we have the veins of the plant, which bring water up from the roots up to the leaves. And then we already talked about the stroma and the thylakoids. So just to review photosynthesis, you guys should know this equation for your exam. You don't necessarily have to know the amount of delta G produced, but you should know that it is positive, meaning that it is non-spontaneous or endergonic. So energy is required for this reaction to begin. And in this reaction, we use solar energy from the sun. 6 molecules of carbon dioxide and 6 molecules of water to form sugar and oxygen. This is also a series of redox reactions, except carbon is reduced and water is oxidized to water and electrons in the form of H minus. The electrons are taken from water as H minus, and they begin as low energy electrons, and they're energized by the light that comes in and eventually passes to carbon dioxide in the form of glucose. So we already talked about the light reactions. This is what we went over in class with photosynthesis 2 and photosynthesis 1. We drew that pathway on the board. If you guys want, I can rewrite that pathway for you as well and post that on Blackboard. If you guys think that will be useful, just let me know. But this was called photophosphorylation. And if we kind of think about it, this makes sense for the name because photo meaning light and phosphorylation, if you remember from our cellular respiration, we talked about that electron transport chain and the phosphorylation that was happening there. And it's kind of this series of back and forth, things are bouncing around and energy's moving and stuff like that. So this is kind of our overview of photosynthesis. So like I said, we have our light reactions, which we've been talking about. And then we have our Calvin cycle, which is our dark reactions. And you can also see really well here how ATP and NADPH is also in this kind of cyclical pathway, same way as our Krebs cycle or just kind of cellular respiration in general. If you remember those molecules, NAD, FADH, and ADP to ADP are all kind of reused. So that's something that our body and plant cells are really good at, is using and reusing materials so we don't constantly have to make new ones from scratch. If we did, that would be a large source of energy just going into remaking these molecules that help us to generate energy, and it would be kind of counterintuitive. So we talked about chlorophyll pigments and kind of their purpose, the way they work. So light is a form of electromagnetic energy. We know this. We talked about the electromagnetic spectrum. We talked about shorter wavelengths having higher energy. So those are going to be like our blue and purple wavelengths are going to be more high energy than our red wavelengths. And pigments absorb certain wavelengths and they look like the color that they do not absorb. Again, why we see green. And we talked about the 3 different pigments for absorbing energy from light that plants have. So this is what I was talking about with the photophosphorylation that I wrote on the board. So this is done by the photosystems, photosystem 2 and photosystem 1. And this is kind of what that looks like. So here we have, over on the left side, so here we have photosynthesis 2, photosystem 2, and we have light coming in and it's bouncing around these chlorophylls. And if you remember the name of them, they were the chlorophyll P680. So the 2 chlorophylls that we talk about are P680 and P700. And so here we have our P680 and we have the light kind of bouncing around and we have it entering this photosystem complex. Just pulling up my notes quickly. So we have the photon coming in to the chlorophyll. And if you remember, a photon is kind of a particle of light. It's very high in energy. And this high energy photon is going to excite an electron, which is then going to be passed around the 680 chlorophyll. And what you're seeing, where's my mouse? Here is an electron from P680 jumping to the primary electron acceptor of PS2. And when that happens, this P680 becomes positively charged because it just lost an electron. So in order to become neutrally charged again, it's going to steal a hydrogen pair from water to make oxygen. So that's why you see here, we have our H2O going in, we have 2 electrons coming off, and we have, you can see here, we have the half of an O2, so we have an oxygen coming off. and this is happening in duplicate. So then if we're following our little electron, he's here in this photosystem, right? So he came off this primary electron acceptor into this photosystem, and he's going to travel now from photosystem 2 to photosystem 1 via an electron transport chain. And this electron transport chain right here is called the cytochrome. And when this happens, we also make some ATP. And while we make ATP, that means we lose energy, right? So if we're taking energy out of our little electron, it is going to be used to make ATP. But now we have another low energy electron going into this photosystem 1. And so our electron comes, but while our low energy electron comes, so comes more light. So our electron's going to become re-excited by another photon. And now this time, this high energy electron is bouncing around the chlorophyll P700. And this is going to go to another electron transport chain, another primary electron acceptor here. And then it's going to go into another primary electron, it's going to go into another electron transport chain. This one is called ferredoxin, and you can see here. And at this point, this high energy electron, you can see here, it passes through this NADP reductase, and we have a hydrogen come off to make NADPH. So NADPH is going to act similarly to NADP, and it's going to be used to continue making energy in the next part of the cycle. So here's just kind of a little bit more in-depth view of the photosystem. So you can see the light coming in, you can see it bouncing around. Here's our P680, our primary electron acceptor, and then again, it's going to go into an electron transport chain. The first one is called the cytochrome, and then the second electron transport chain is called ferredoxin. So here's kind of the play-by-play. Light comes in, right, energizes our electron. We bounce around, we go to P680. And when we get to P680, we're going to go to the primary electron, the primary acceptor. And here's the oxygen being made, water coming in, being used. Now we're bouncing around our cytochrome complex, our electron transport chain. We lose some of our energy to make ATP. We come back in, we go to the P700. Here's more light coming in to re-energize our little electron. We go into our primary acceptor again and down another electron transport chain. Again, this one's ferredoxin. And you can see here the FD. And here we have more, we're losing more of our energy. We're losing some of those hydrogens, right? Because our electrons are moving around in the form of negatively charged hydrogen molecules. So here we're making NADPH out of when we go through this NAP plus reductase. So here's just another view, a cute little animation for you guys, another way to think about it. So you can see here, this energy that's, this work that's being done, I should say, is essentially a photon. So this photon is giving energy into the system. It's giving this little charged electron the energy to go all the way up here to this woman. And this is in photosystem 2. So remember, annoying, I know, but photosystem 2 happens before photosystem 1. Again, we like to make things confusing in science. So we're here, right? We got the energy from this photon. We are charged. And we kind of slowly go down this, they're calling it this mill. This is our cytochrome. And here we're losing some energy to go and make ATP. Now our tired low energy electron is going back here, getting re-energized by more work from another photon, more energy from another photon. It's getting elevated again, and then going to this NADPH final product. So as I just said, this is linear electron flow. So now we're going to start getting into some new stuff for the day. So we have that was the linear flow of electrons. This is the cyclic flow of electrons. So cyclic flow is a short circuit, quote unquote, of the regular linear pathway of electrons. And this only happens in photosystem 1, where there's no splitting of water and no oxygen produced. So electrons are passed in a cycle. So we go from cytochrome C to P700 to the primary acceptor to ferredoxin, remember another electron transport chain, and then back to cytochrome C instead of to NADPH. And when this happens, we generate ATP, but no NADPH. And what is the purpose of this is that the Calvin cycle consumes more ATP than NADPH. So in the linear flow, which we've been talking about, the same amount of NADPH and ATP is produced. But in cyclic flow, we use this to make up the difference, and this gives us a little boost of ATP. So this is kind of like another, not necessarily another step, but it kind of happens in place of that linear flow that we were talking about. So it's just another kind of caveat to photosynthesis in general. So again, this is our linear flow model that we've been talking about. And now this cyclic flow is another way for us to generate ATP. And again, the reason for this is because the Calvin cycle, that dark reaction cycle, which we're going to talk about next, consumes more ATP than NADPH. So we being plants now, need to make more ATP in order for the Calvin cycle to work properly. And this is the way that we do it. So if I were to ask you a question differentiating between linear flow of electrons and cyclic flow of electrons, you would say here, or if I asked you compare and contrast to, I guess would be a better way, you would say that they both involve cytochrome, they both involve P700, they both involve the primary acceptor, and they both involve ferredoxin. In cyclic flow, the electron goes back to cytochrome C, and in linear flow, it goes into the NADPH reductase. Cyclic flow generates ATP, linear flow generates NADPH. So kind of a good question, right? Differentiating between the two might be a good thing to know. Again, all of this is going to be on your exam, so I'm sure that this is going to come up in one way or another. So make sure you are studying this. Okay, so ATP production in photosynthesis is by chemiosmosis. Just like in respiration, the electron transport chain pumps hydrogen from the stroma into the thylakoid space to build a gradient. Hydrogen flows back down the gradient through the ATP synthase. So this is exactly like what happens in cellular respiration with the ATP synthase and that gradient pump. We called it oxidative phosphorylation in cellular respiration, but very, very similar. We also used the chemiosmosis in cellular respiration. Sorry, the word completely escaped me. And I had a question on what is chemiosmosis. And so I just wanted to give you guys the definition of that, is that chemiosmosis is the movement of ions across a semi-permeable membrane-bound structure down their electrochemical gradient. And the example, obviously, is the movement of electrons or hydrogen ions to form ATP through the ATP synthase. So again, that definition for you, and you can also find this in your textbook if you guys ever need a word defined. The back of your textbook has a glossary. But chemiosmosis is the movement of ions across a semi-permeable membrane-bound structure down their electrochemical gradient. So if you remember in our oxidative phosphorylation in that electron transport chain, we have the H plus molecules being pumped up to form that gradient. And then the chemiosmosis is when they come down the electrochemical gradient through the ATP synthase. And that's where, again, ATP synthase is where our ATP is synthesized. So same thing, photosynthesis, right? Chemiosmosis, we have an electron transport chain pumping the hydrogen ions against their concentration levels, right? So we're making a gradient. We're going from low concentration to high concentration. And the reason we're doing this, again, is to build that gradient so that hydrogen ions can then flow down the gradient and through the ATP synthase. Okay, so you can see here, we have our hydrogen ions coming down through this ATP synthase, and here we're making ATP, okay? And we're also, some hydrogen ions are still coming through, okay? But this is, the thylakoid space is an area of high H plus concentration. The stroma is low H plus concentration. So this is where our gradient effect is coming in, right? High to low. And when we go through that, our ATP synthase is, of course, cranking out some ATP. So next, we're going to discuss the Calvin cycle. And the Calvin cycle is where we use our ATP and our NADPH to synthesize glucose. And this is called the dark reaction because we're not using photons in this step. So we do still use the products of light reaction, similar to how the citric acid cycle is anabolic and not but instead of being an anabolic reaction, this is a catabolic reaction. Sorry. So Calvin cycle, catabolic. The Calvin cycle happens in 3 phases. So first we have our carbon fixation, and this is performed by the rubeose. This is the most abundant protein in chloroplasts and possibly in Earth, because if you think about how many plants there are, right, on Earth, so all types of plants, including algae, they all have, some of them are unicellular, some of them have a gazillion cells, and inside those cells are multiple chloroplasts, okay? So this is a protein found in chloroplasts, and it's not just one per chloroplast, it's many. So that's what makes it the most prolific protein possibly on Earth. Anyway, carbon fixation. So CO2 is added to RUBP, which is the name for the carbon acceptor. And this fixation happens again by this rubeose. Next, we have the reduction, which is where we add electrons, again, in the form of H minus from NADPH. So this H is going to come off and remake the NAD plus. Finally, we have the regeneration of the carbon acceptor, RUBP. So I just want us to watch this video. So this is going to kind of give us an overview.
Professor Dave here. Let's learn about photosynthesis.
He knows a lot about the science stuff. Professor Dave explains.
We talked about how cellular respiration transforms the food we eat into ATP. But what do plants do? They don't really eat in the normal sense of the word, apart from Venus flytraps. As it turns out, plants are getting their energy directly from the source, the sun. Plants use sunlight as well as water and carbon dioxide, which are plentiful in our earthly surroundings, to produce their own nourishment through a cellular process called photosynthesis. This word makes sense because photo comes from the Greek word for light, and synthesis means to build. So plants use light and small molecules to build bigger molecules that they can then metabolize to produce energy. Other organisms then eat the plants and other organisms eat those organisms. So plants really are the foundation of the entire food chain. Let's learn about how photosynthesis works. Photosynthesis consists of two stages. There are the so-called light reactions, in which solar energy is converted into chemical energy. And then there's the Calvin cycle, which uses that chemical energy to build G3P, the precursor to glucose and other molecules. Both of these stages occur inside one of the organelles found in plant cells, which are called chloroplasts, and there are around 30 to 40 of them per cell. A chloroplast has two membranes that surround a fluid called stroma, within which sacs called thylakoids are suspended. These sacs are often stacked in columns called grana, and in the membrane of these thylakoids sits a special pigment molecule called chlorophyll. Chlorophyll can take either an A or a B form, differing only in one functional group inside the porphyrin ring, either a methyl or aldehyde group. The porphyrin ring is what gives this molecule the capacity to absorb sunlight in a unique way. If chlorophyll absorbs a photon of a particular energy, one of its electrons jumps up to an excited state. These chlorophyll molecules sit inside of a photosystem with many other chlorophylls, and this excitation can get passed around from molecule to molecule, with one electron relaxing back to the ground state, emitting another photon that will excite an electron in another molecule, and so forth, like a pinball machine. Eventually, a photon may strike the reaction center complex, which also contains two molecules of chlorophyll. When this happens, the high-energy excited electron, rather than relaxing back down to the ground state, will instead be transferred to another molecule called the primary electron acceptor, after which an enzyme supplies the missing electron to the oxidized chlorophyll via a water molecule to get it back to normal, which is how oxygen molecules are produced. This is the first step of the light reactions. These are a series of redox reactions that occur first in photosystem 2 and then in photosystem 1. If they seem like they are named backwards, it's because they were named in the order that they were discovered. In photosystem 2, electrons flow beginning with the primary electron acceptor through a series of compounds that are embedded in the thylakoid membrane, sort of like the electron transport chain from cellular respiration. ATP will be a byproduct of this process due to a proton gradient and ATP synthase, also just like in cellular respiration. Now electrons will move through photosystem 1, another electron transport chain. But instead of producing ATP, photosystem 1 will result in the conversion of nicotinamide adenine dinucleotide phosphate, or NADP+, into NADPH. So the light reactions in the thylakoid, which are summarized by the following equations, require sunlight and water among the reactants and generate oxygen, ATP, and NADPH as products. ATP and NADPH are then used in the Calvin cycle, which occurs in the stroma. This is where all the synthesis occurs. Unlike the citric acid cycle, which is catabolic, the Calvin cycle is anabolic, building organic molecules from smaller components, requiring energy to do so. This happens in 3 phases. Phase 1 is carbon fixation, which is catalyzed by an enzyme called rubisco. Rubisco captures CO2 from the atmosphere and attaches it to a 5-carbon sugar called ribulose bisphosphate. The resulting 6-carbon molecule is unstable and splits into 2 molecules of 3-phosphoglycerate. Phase 2 is reduction. Each of these molecules receives a phosphate from ATP and is then reduced by NADPH, after which it loses a phosphate to become glyceraldehyde 3-phosphate. Some of this G3P is the output of the Calvin cycle, going on to other pathways that generate glucose and other organic compounds. And some goes back to generate more RUBP and start the cycle over again. To generate a net output of 1 G3P, the cycle needs 9 ATPs and 6 NADPHs. So to summarize, photosynthesis occurs in 2 phases. In the light reactions, light and water go in, and oxygen comes out, as well as the ATP and NADPH that are used in the Calvin cycle, where carbon dioxide comes in and organic products like sugars come out. In this way, it's almost like the reverse of cellular respiration. They involve a series of redox reactions. The electrons just flow in opposite directions. In one, a sugar is built. In the other, a sugar is degraded. And that's how plants make their own food. Thanks for watching, guys. Subscribe to my channel for more tutorials. And as always,
Okay. So that was a good little introduction into our citric acid cycle. Okay. Is this the right? Sorry, not the citric acid cycle, the Calvin cycle. Where did it go? Running away from me. Stop share. Okay. No, what is happening? Stop sharing. Share screen. Hang on 1 second. Sorry, some technical difficulties. Let's see. Okay. It wouldn't be a virtual class without some technical difficulties anyway. Okay, so that was a good review of the Calvin cycle. And now we're going to get into it. So again, we have another one of these circle cycles. And we have some big words again. I know they're scary. But thankfully, some of these words are actually fairly similar, right? Our 1,3 bisphosphoglycerate, phosphoglycerate, and stuff like that. ATP we've seen before, NADPH. So it's really very similar to what we've been learning about. It's just another one of those where you have to go in and you just have to write out the cycle a few times and really just commit it to memory. So we have the input of 3 carbon dioxides entering 1 per cycle. So we have our phase 1, our carbon fixation. And here we have our 3 phosphoglycerate. We have 6 ATP come in and they go to 6 ADP. So our 3 phosphoglycerate goes to 1,3 bisphosphoglycerate. Next, we have 6 NADPH come in. So we're adding 6 hydrogens and we're taking off 6 phosphate groups in our reduction phase. This makes G3P, okay? Our G3P comes to the output, and this is what we use for glucose and other organic compounds. So some of it's coming out, some of it's still going around, right? So here's our GP3, and this is our regeneration of RUBP. So how do we regenerate the RUBP? We have 3 ATP molecules go to 3 ADP molecules, and that makes, when we add those phosphate groups, we go back to our RUBP, okay? That's our Rubisco. So again, we have our carbon dioxide coming in, going with our Rubisco. Then we have the 3 phosphoglycerate. We put 6 ATP into this, and the 6 ATP here go to the 6 ADP. So now we have the addition of our 6 phosphate groups, right? We have our 1,3 bisphosphoglycerate. We add our 6 hydrogens from NADPH, and our 6 phosphate groups come off. This is our reduction phase. So now we have this 6 G3P, 1 GP3 comes out for the output. The other, the 5 GP3, comes, gets we add those phosphate groups back on again, right? And we make our RUBP. Same thing, keeps going around in a circle, okay? And then you can see here how when our NADP comes out and our ADP comes out, this allows us to reuse those molecules back in our light reaction again to again make ATP and NADPH. So certain plants have some special adaptations, right? We kind of talked about this when we watched the Amoeba Sisters video about the cacti and how they open their pores at different times. So for our sugarcane, we have the C4, and this is all happening during, you can see the mesophyll cell and the bundle sheath cell. So these are some different cells, and this allows for spatial separation of steps. Whereas with the pineapples, we have the temporal separation of steps. So while this is happening in different cells, this is happening at different parts of the day or night. So during the nighttime, our pineapples are opening up their pores to allow carbon dioxide to come in, oxygen to go out. And then during the day, they're using that carbon dioxide for this Calvin cycle. Whereas for sugar canes, this is again happening in different cells. So in this way, our pineapples are kind of acting like our cacti did. So these are some review slides that you guys are going to be able to go over, or review questions. I also want to bring your attention to try sharing another screen again. So this is obviously our Blackboard. Here you have a review sheet for exam 4, along with some short answer reviews. Again, these are from another professor, so they're not necessarily completely indicative of what our text is going to be like. But that being said, it's going to be fairly similar because of there's only so much information that we can really put on the exams. So that being said, that is the end of kind of our new lecture material. I do have some stuff that some of the students had asked me to go over in class, so I'm going to present that now. But if you're done watching for the day, I did say I was going to give you a question that you had to email me the response to. And the question is that I want you to email me is the name of my cats, because I can't think of anything else. So their names are Sushi, Sashimi, and Mochi. And they are the cutest things ever. So I can show you guys a picture of them in class on Tuesday if you're interested. But that's going to be your question that you're going to email me for your attendance. Again, because this is a virtual lecture, I'm giving you guys until Tuesday of next week to email me that. So that'll count as your attendance, like I've been saying. And but since it's a virtual lecture, I figured you don't really have to watch it today. I recommend you watching it a couple of times because it's very helpful if you watch things over and over again for you to remember them. Again, since this will be on the exam, you really should watch the lecture. I don't really know what else to tell you. Anyway, so if you think you know the glycolysis, the Shun step, and the Krebs cycle, and you don't want to go over it again, totally fine. Watch those videos, keep writing out those pathways, and don't forget to study other things too that we talked about. All these multi-step processes are important, But don't forget things like the redox reactions and kind of some of that terminology from our photosynthesis unit. So, and this is really nothing new. This is what we've been seeing. It's just written out in a different, I just kind of tried to reword things again to see if maybe this sticks a little bit better with anyone. So we have, oh, okay. So we have our first step, right, in cellular respiration is glycolysis. So what happens in glycolysis is we have a phosphate group added to glucose. So we're starting with glucose. And this makes glucose 6-phosphate. When this happens, we have one molecule of ATP go to a molecule of ADP, right? Because we're taking a phosphate group off of the ATP and adding it to the glucose. Next, this glucose 6-phosphate goes through a rearrangement to make fructose 6-phosphate. We add another phosphate group. So again, we have ATP going to ADP, and that phosphate group is being added onto our molecule. This makes 1,6-bisphosphate, 1,6-fructose, fructose 1,6-bisphosphate. This molecule then breaks apart into 2 3-carbon sugars. So we have 2 molecules of ATP coming in to work on 1 glucose molecule. When this molecule splits, everything here, you can see here, the products are happening in duplicate. And this is why we end up with a net gain of ATP. So we start with 2 ATP, but we make 4 ATP. So we have a net gain of 2. So anyway, our fructose 1,6-bisphosphate breaks apart into these 2 3-carbon sugars, and these 3-carbon sugars rearrange to make glyceraldehyde 3-phosphate. This is then oxidized back into 1,3-bisphosphate bisphosphoglycerate. And when this happens, so remember it's happening in duplicate, NAD is going into NADH, and ADP is going into ATP, okay? So this is happening when glyceraldehyde 3-phosphate oxidizes. We're losing a hydrogen and we're losing a phosphate group in duplicate. So it's happening twice, okay? So, nice. Sorry, I just wanted to make sure I got that written right. Okay. Next, the phosphate, we transfer a phosphate to ADP. This phosphate moves to make 2-phosphoglycerate. Then we have a lysate reaction, remove water to make phosphenylpyruvate. Then we have another kinase reaction, removing a phosphate to make pyruvate. So again, here we have ADP being made into ATP. And this is where our next 2 ATP come from, and our final product, the pyruvate, okay? So now we're taking this pyruvate from glycolysis and we're putting it into the shunt step. Again, remember glycolysis is occurring inside of the cytoplasm or the cytosol, and the shunt step is now going to bring that pyruvate from the cytosol into the mitochondria. So it goes through a transport protein, and as soon as it enters the mitochondrial matrix, a carbon dioxide comes off the pyruvate, and then hydrogen from NADH comes off as well. So we are making NADH. When this happens, we also have the addition of coenzyme A, and this forms acetyl-CoA, which is the molecule that then goes into the Krebs cycle, okay? So there's our shunt step, CO2 comes off, and a hydrogen comes off, CoA goes on, and now we're in the Krebs cycle, Okay, so remember, this is another cyclical cycle. And the reason for that is because this end product, the oxaloacetate, is also being used in this first step. So it's going to go from step 8 back into step 1. So we have our acetyl-CoA and our oxaloacetate combined to form citrate. Our citrate then gets transformed into isocitrate. Isocitrate forms alpha-ketoglutarate by CO2 coming off and NAD going to NADH. Okay, so we have a carbon dioxide and a hydrogen coming off of isocitrate. That leaves us with the alpha-ketoglutarate. The alpha-ketoglutarate binds with coenzyme A. Another carbon dioxide comes off and another hydrogen comes off. Okay, so now we have our succinyl-CoA. Succinyl-CoA, the coenzyme A comes off, and we transform GDP to GTP. I think I wrote that backwards. Oh no. So we transfer a phosphate group to GTP. Okay. Now we have GTP, and that's going to become GDP because that phosphate group is going to immediately go from this GTP over to ADP. So we're making ATP. Okay. So we have succinate now. Succinate, we're going to lose a hydrogen to FADH, and we're going to make fumarate. Fumarate, we're going to add water to make malate. And then from malate, we're going to lose another hydrogen to go back to this oxaloacetate. And that oxaloacetate is then going to go back into step 1 again. And someone asked kind of like, what is the purpose of all of this? Obviously, you guys have asked me multiple times, why do we need to know this? But the reason for all of this, the reason for glycolysis, for the Krebs cycle, and then also remember, we don't stop the Krebs cycle. After the Krebs cycle, we go through the oxidative phosphorylation with the electron transport chain to make that hydrogen gradient, and then have those hydrogen molecules flow back down through the gradient through the ATP synthase, right? So the whole point of cellular respiration is to make ATP. Okay, that's the whole point of it. So and again, we've talked about why we need ATP. ATP is what our cells use for energy. If we don't have ATP, all of our cellular functions will cease to exist. Okay. The interesting thing about glycolysis, right? We have to remember that this can also be done anaerobically. And if we are using anaerobic respiration, we are also doing fermentation. So remember, we talked about the 2 different types of fermentation in the cellular respiration chapter, the lactic acid fermentation and the alcohol fermentation. So you guys need to know those as well. If you remember, they're kind of simpler cycles, and while they're great for that time when we have low oxygen, you cannot just sustain life off of no, I lost the other PowerPoint. You can't sustain life just off of fermentation cycles. Make sure you guys can see the right slide. Okay. So just to quickly review again, so our alcohol fermentation converts pyruvate to ethanol and carbon dioxide, and this is happening in yeast. So this doesn't happen in humans, okay? We are not making alcohol fermentation. We are making lactic acid fermentation. And in lactic acid fermentation, we're converting pyruvate to lactic acid. Okay? This is the pathway for that. So again, we're using our pyruvate, so our end product of glycolysis, and rather than having it go into the shunt step and into the Krebs cycle, it's just being reused to replenish the NAD production. Okay? So again, if you remember, if we lose oxygen, everything stops, right? If there's no oxygen, we can't hand off hydrogens to our, if there's no oxygen, yeah, we can't make any of the byproducts of cellular respiration that we need to. So everything stops. The final acceptor of the electrons is oxygen to create the water molecule. So if there's no oxygen, there's no electron transport chain happening. If there's no electron transport chain happening, there's no gradient of hydrogen ions for the ATP synthase. So no chemiosynthesis. And NADH and FADH2 can't pass off those hydrogens. So there's a buildup of NADH and there's no NAD plus or FAD plus. So the reason why we use anaerobic respiration is to replenish those NAD plus molecules. So we can still have some sort of energy production happening. Again, glycolysis only makes a net of 2 ATP. The ATP synthase makes like 30 to 32 ATP molecules per cycle, I believe. So we're obviously experiencing a huge energy deficit based off of when we can do aerobic respiration. And then again, also as a reminder, while we talk about this starting with glycolysis and starting with glucose, that's not always the case, right? If we ingest carbohydrates, we have these large macromolecules that need to be digested into simpler sugars so that we can make glucose and start from glycolysis. But other parts of our diet, such as proteins or fats, so amino acids, glycerol, fatty acids, also can enter this cellular respiration cycle at different parts. There's another cellular respiration video for you guys if you're interested. So yes, that's pretty much everything. This test is only on 2 chapters. Please come ready to ask questions on Tuesday. We're going to be doing our last review before the final. And then you guys, because your final is later, I'm sure you probably have the chemistry final the same day, knowing how the rest of the semester is kind of panned out. But if that's not the case, you guys have plenty of time to study for this final. So I cannot suggest enough to you that you use all the time available and you really, really study well. Again, this exam cannot be dropped. So really put your best foot forward when it comes to this exam. The other thing is, and I'll talk to you guys about this on Tuesday as well, but if someone were to come down with COVID, there's a special procedure that has to be followed. And I'm going to get you guys the information for that on Tuesday. But yes, if you have any other questions, please feel free to email me. I'll be checking my email over the weekend and all day tomorrow. And I'm also going to make sure I post those chapter packets for you guys. I'm going to do that right now. So those will be under the 2 chapters that our exam is on. So that's all for today. I hope you guys have a great day, and I will see you all on Tuesday. Have a good weekend. Bye.